Hello class 10th. Let's continue chapter 1, cell cycle, cell division and structure of chromosome. In previous video we discussed about the cell cycle, cell division, two types of cell division we discussed, mitosis and meiosis, right? Now today in this video we will discuss about the structure of chromosome. All of you know chromosomes, they are what? These chromosomes are seen in the nucleus of dividing cell only at the metaphase and anaphase stage of cell division, whereas in non-dividing cells they are seen as they are seen in the form of a network of thread-like structures, and what they are called chromatin reticulum. Right now, the question is, what is chromosome? Chromosome is a thick rod-like structure bearing genes present in the dividing nucleus. The number of chromosomes always remain constant in the cells of each species. All of you know in human beings, how many number of chromosomes are there? There are 23 pairs of chromosomes. Right? Depending upon the number of chromosomes, the cells are said to be haploid or diploid. Haploid means a cell. What is haploid cell? A cell in the nucleus of which each type of chromosome is one in number. Right? And diploid cell is a cell in the nucleus of which chromosomes are present in the form of pairs. Now next is here, what are homologous chromosomes? Two chromosomes of one pair which are exactly similar and bear same genes at same loci, means same position, then the chromosomes are called homologous chromosome. Okay, now next is your structure of chromosome. Now I am attaching a video through which you can easily understand the structure of chromosome and their types, right?
We know that the long stretch of DNA is coiled to a great extent to fit inside the tiny nucleus. This process of coiling gives rise to the chromatid structure. It's a thread-like mesh that we find in the nucleus of a metabolically active cell. The chromatids gain a typical classical structure which looks somewhat like this. It's seen during cell division. And these are nothing but the chromosomes. But tell me, do all chromosomes look exactly like this? Do all the 46 chromosomes look the same? If yes, then why do we say that there are 23 pairs of chromosomes? What helps us categorize these chromosomes as pairs? Well, all chromosomes are not exactly like one another. They come in types depending upon their structure or shapes and functions. The first basis for classification is shapes. We know this is the structure of a typical chromosome. And is this a single chromosome? That's right. The difference is that this chromosome is not duplicated, while this one has a duplicated copy attached to itself. So these are the duplicated sister chromatids which are attached to the same chromosome. Now to understand the types, let's first understand the typical structure. Here, these two regions are called the arms of the chromosome. This arm is named as the P arm and this is known as the Q arm. So how do we recognize which is the P and which is the Q arm? Is it based on their positions? Nope, it depends on the size. The smaller arm is the P arm while the larger one is the Q arm. So this chromosome has one P and one Q arm, while this one has two P and two Q arms. Now let's focus on this chromosome structure. Can you guess what this structure is called to which the arms are attached? Well, this knot-like structure is called centromere. It's the portion of DNA that acts as a link between the two sister chromatids. But that's not its sole purpose. Its major function is the attachment of spindle fibers during the cell division process. And let me tell you one more interesting use of the centromere. It's the position of the centromere that helps us categorize the chromosomes into 4 types. The four types include metacentric, submetacentric, acrocentric, and telocentric chromosomes. Let's have a look at the structures of each type. Aren't these simple to understand? This one is called metacentric type. As we can see, the centromere is placed almost exactly at the center. The two arms seem almost of the same length. We can say that the centromere is placed somewhat in the middle of the complete structure. Now the next type is the submetacentric. Here, the centromere is placed slightly away from the center. As a result, the two arms of the chromosome appear unequal. The next type is the acrocentric. In this, the centromere is seen almost towards the end region. The term acro in Greek relates to peak. Thus, the name acrocentric refers to the type where the centromere is near the terminal region. Lastly, what we see is that the centromere is present at the extreme end. That is, the centromere is placed at the telomeric region. Hence the name telocentric. However, these chromosomes are not found in humans. The human karyotype lacks telocentric chromosomes at the other type of classification. This is a much simpler type of classification. Here, the chromosomes are classified whether they are autosomes or sex chromosomes. Now, what could these be? This is a representative image of a typical human karyotype. The chromosomes that pair from 1 to 22nd are called autosomes. That is, they are not the sex chromosomes. To be precise, they have got no function to deal with the sex of the individual. The last pair, however, determines the sex of the person. So if the two are identical, that is, both are X and X, then the individual is a female. And what if both are different? In such a case, when one chromosome is X and the other is Y, the individual is a male. This pair is the sex chromosomes. So the chromosomes which help in determining the sex of an individual are sex chromosomes, or even called allosomes. The others, which are from 1st to the 22nd pair, are not the sex chromosomes. This makes them the autosomes. Both contain genes that code for proteins which make up our body.
DNA, or deoxyribonucleic acid, is the heredity material of the cell. Heredity is the information that passes from one generation to the another. Most DNA is present as linear double-stranded helical structure in nucleus. DNA double helix is made of sugar phosphate backbone with nitrogenous base or nucleotides at the center. The two strands run antiparallel and are linked by complementary base pairing of bases through hydrogen bonds. That is, A pairs with T through double hydrogen bond and G pairs with C by triple hydrogen bond. The sequence of four bases, that is A, T, G and C, codes for the genetic information of an organism. In an organism, each cell has the same genetic information, that is, they have the same DNA. Each human cell has 46 DNA molecules, 23 from each parent. The human genome comprises of 3.2 billion bases. The human DNA has about 20,000 genes which encode different protein or RNA. Gene is the unit of heredity. Gene are the DNA regions separated by intergenic regions. The total DNA of human cell measures around 2 meters if fully extended, and if total DNA of all the human cells are summed up, its distance would be equal to 70 complete trips from Earth to the Sun. So how this 2 meter long DNA can fit into the tiny nucleus? The answer is through DNA packaging. DNA folds and packages with the help of histones and accessory proteins to form a complex and very compact structure called as chromosome. Histones are positively charged basic proteins that binds to negatively charged DNA. The genetic information or DNA is transferred from one generation to another in the form of condensed chromosome during cell division.